All right, hitting it now.
Hi, everyone. Welcome to our live lesson on COVID-19 testing. We'll be talking about how the PCR test to detect the SARS-CoV-2 virus works. We'll also address some common questions and misconceptions. And then at the end, we'll get to your questions about testing, vaccines, and more. So this lesson is designed especially for high school students, and it's part of a larger case study that our team published last year, where you can learn about clinical lab careers and even interpret your own PCR test results. So we're so happy to be here live with you. We'll be going through this presentation that we're sharing, but also welcome your questions and comments. So if you're watching live right now, please feel free to write in the chat box that you should see on your screen, and we'll try to respond to everyone at the end and maybe along the way as we monitor.
Awesome. Well, before we get started with the science, we wanted to share a little bit about ourselves. I am Dr. Emily Pietzu, and I'm joined by my colleague, Dr. Christina Valianatos. And we're coming to you today from the Jackson Laboratory, which is a non-for-profit research institute. And I put a couple pictures of our campuses up on this map for you here. So the Farmington campus is depicted on the left there. That's where Christina's home base is. And on the right is the Bar Harbor campus of the Jackson Lab. That's where I'm based. So we're both science educators, and we work in the genomic education division at JAX. We were both originally trained as research scientists, so we have our PhDs. I've got mine in genetics and genomics, and Christina's got hers in human genetics. So we're very passionate scientists, but we're really excited to now be working in education to bring science to life and connect students with careers and opportunities in the STEM field.
Thanks, Emily. So today we'll be going through the steps of the COVID PCR test, and then we'll analyze some data together to see how the test results are determined. And then at the end, we'll address your questions, like I said. And so don't forget, this is part of a larger lesson that our team developed called The Power of a Test, COVID-19 Diagnostic Testing, and Who Does It? We published this in the National Centers for Case Study Teaching and Science. And you can do this lesson at your own pace anytime. It's freely available for students or for teachers on our own JAX website, JAX Education website, or from the National Centers for Case Study Teaching and Science. So be sure to check out the entire thing. Okay, let's dive in. Let's start with the lesson that we're going to do today.
All right, I'm ready. All right, so this lesson follows the story of Marcus. He's a recent college graduate, and his job is as a technician in a clinical lab. And he took this job to gain some research experience for a year or two before applying to medical school. And as the story goes, Marcus's work shifts unexpectedly as the COVID-19 pandemic starts to unfold, and he has to begin running PCR tests for SARS-CoV-2.
So we know that a lot of students out there might be familiar with working in a kind of traditional research laboratory, but we want to show you what's the difference between a traditional research lab and a clinical lab.
Well, I feel like there's lots of similarities, right? So both do research, but in a clinical lab, they have to undergo some special certifications to work with human samples and be able to reliably perform diagnostics. But, you know, there's lots of career opportunities from entry-level assistants like Marcus to lab managers, doctors, and we won't be going over these career opportunities specifically today, but in the full case study, there are 2 sections on careers, and we welcome you guys to browse those later.
So in this lesson, we're going to go over how the COVID-19 PCR test works and how a diagnosis is made. So this is perfect if you've ever gotten one of these PCR tests and wonders what exactly happens after they do that swab and take your sample away.
Okay, so before we get started on the testing, let's just talk about these zoonotic diseases for a second. So this is a term that generally describes diseases in humans that are originating in animals. And if you look at this picture, there's a lot of diseases that we live with today that have their origins in animals. Things like Lyme disease, we can see tuberculosis on there, HIV and Ebola. these all originated in animals before spreading to humans. So we just wanted to remind you that studies suggest that SARS-CoV-2 also originated this way and spread to humans through close contact.
Okay, so what is this SARS-CoV-2 virus? So it's the name given to the coronavirus strain identified in this current pandemic that we're in. It has a specific name to identify it because it's just one of many types of coronaviruses that exists. So think of any species like dogs or monkeys or humans. It's one species with many individuals that share a lot of common traits, but also each are unique. So coronavirus is a group of viruses. They all have spike proteins on the outside, shown here in that picture in red. And they resemble this crown-like structure, thus the Latin word corona in the name. And viruses, just like plants, animals, and people, they have a genetic code that's the blueprint for making them how they're supposed to be. So when we're designing a test, how can we specifically identify each coronavirus and distinguish SARS-CoV-2 from other viruses? So the answer is the genetic code. And that's what this figure shows here, how the genetic code for the SARS-CoV-2 virus encodes different parts of that whole entire virus. In this case, the virus has an RNA genome. So its genome is RNA and not DNA, and that's going to be important in a minute. So let's see exactly how we do this genetic test.
All right. So I guess many of us have taken these PCR tests by this point, right? So you're familiar with going to the testing site, which is, actually in the clinical setting referred to as a sample collection site. And they take the swab of your nose, and then what happens? Well, the sample is sent to the clinical lab to follow the diagnostic testing protocol. And here it is. So for this genetic test, there's 4 major steps, from the lab work to data analysis and interpretation. We're going to go through all of them in detail together. So let's get started on this lab work.
But first, to do any sort of test, we need tools to do all these steps. So if you've done research before, either in class or in a research lab, you'll notice that the equipment used in the clinical lab is actually very similar to a basic research lab.
That's right. Check out all of this equipment you might be familiar with already. After the individual patient sample arrives at the diagnostic lab, they're processed in these small tubes and then stored in tube racks. Micropipetters are used to draw up liquid solutions used in the various steps of the protocol. And multiple samples can be handled in parallel together on a microplate to save time. These plates can be loaded into 96 different samples can be loaded onto this plate and then put into the PCR machine, used for amplification and detection.
So the very first step of the diagnostic testing protocol is nucleic acid extraction. So we need to extract the genome of our target. Since it's a genetic test and we're looking for the genetic sequence of the target, in this case, the SARS-CoV-2 virus, we need to isolate the genomes collected in our swab. So let's take a moment to think about that nasal swab that was the patient sample. What exactly is in that sample?
I'm thinking. Well, we're trying to detect the virus, but that's not the only thing that's in your nose, right? We know that there's bacteria that are part of our microbiome. Maybe you have another virus like a rhinovirus if you have a cold. And of course, we have our own human cells in there from our nasal epithelium. So when we take a swab of our nose, we expect to find a mixture of viruses, maybe some bacteria, and definitely some human cells, right?
Yeah, exactly, Emily. So we can't tell what we have in that sample just after the swab. So we're going to isolate the genetic material from everything that was collected in that sample. And that also means we're going to have a mix of genetic material. So think about what those cells are. So both human and bacteria cells, they make both DNA and RNA, right? And depending on the virus that might be in that sample, it could have a DNA genome or an RNA genome. So we'll do a protocol to lyse the cells or open them up to expose all of the nucleic acids present, so all of the genome. Excuse me, I'm in a conference room, so got to turn the lights back on. So we're going to do the protocol to lyse all of the cells and open them up to expose all the nucleic acids. But if we have all these different cells, how do we isolate only the RNA and only the RNA from the virus?
Well, that's a really good question, Christina. When we started writing this, we didn't really know the answer. So how can you actually differentiate the viral RNA from all the rest of the RNA that might be in there? Well, there is a way to isolate it, and it's based on the size, so the molecular weight. So the SARS-CoV-2 genome, it is RNA, and it's about 30,000 base pairs long. And the typical human or bacterial RNAs are going to be in the range of a couple thousands or a couple hundreds of base pairs. So because of this, we can actually use a protocol that selects for RNA based on size. And then you've got your viral RNA ready to analyze.
Right, so it's important to remember some limitations from this part. So you're only going to get viral RNA if it's actually there in your swab sample to begin with. And if you are infected, that means the virus is replicating inside of you, but as time goes on, you're going to get more and more copies. So again, depending on like when the timing of the testing is done, your sample might be different, and then the results from that nucleic acid extraction might be different.
All right, so if the SARS-CoV-2 genome is RNA and we have RNA in our sample, what's the difference between RNA and DNA, Christine? I don't remember. I know there's something about the chemicals, so let's see if this picture can help refresh my memory. That's right. RNA has that hydroxyl group right here. That's the difference. So RNA has this hydroxyl where DNA has this hydrogen. And this makes it much more reactive and prone to degradation. Plus, we know that RNA is single-stranded, right? That makes it a lot less stable and trickier to work with. So that's why basic biologists and chemists are really, really important for doing research, because we use this fundamental knowledge to develop laboratory tools and techniques. So because we know DNA is better suited to be replicated and stored, we can convert RNA to DNA in the lab so that we can work with the RNA in the sample and perform the test. This process is called reverse transcription. Christina's going to tell you a little bit more about it.
Yeah, because this term is probably a new term for you. So let's walk through it. You're likely familiar with the central dogma of biology. So this is where DNA gets transcribed to RNA, which gets translated to proteins. But reverse transcription is a technique developed by scientists to take RNA and make complementary DNA copies, or cDNA, and that's depicted here in this photo. So this doesn't happen naturally in our human bodies, but actually some RNA viruses use this technique to replicate. And this is how scientists first learned about it and developed it into a tool that we can use in the laboratory. And that's how we're using it now.
Very cool. So how does this actually work? It's really cool that we can use RNA by first turning it into cDNA, but how does the reaction get set up and what are the different components? So let's take a look at how it works. All right, so that viral RNA that we just isolated in solution from step 1 that came out of our sample, that's depicted here in step 1. Now, the solution that we're going to put this RNA into contains short-stranded DNA oligonucleotides called primers, and these primers are going to bind specifically to the RNA based on sequence. The solution also contains free nucleotides, those A's, C's, T's, and G's that you know about. And it also is going to contain the reverse transcriptase, that really special enzyme that Christina was just telling you about that's going to be able to convert the RNA into DNA. All right, so after we've got all those components mixed up in the tube, the RT enzyme actually uses that primer to begin adding on nucleotides to the complementary RNA. The enzyme works along the template, reading the sequence, adding in the complementary nucleotides, until it reaches the end and it makes a full copy of that RNA template. And it's going to do this for all of the RNA that's present in that solution until the reaction comes to completion and we have a full cDNA library that's now ready for amplification and detection by PCR.
But this isn't just any PCR that we're talking about. This is quantitative PCR. So it has a dual purpose to both amplify that DNA that you just generated and also to detect your target. So this is another reaction that needs very specific reagents, each with its own purpose. if you're familiar with PCR or polymerase chain reaction, you'll notice that this is a really similar process. So you'll add that cDNA, which you just created, and you want it amplified and you want it detected because that's your template, that's your target that came from your human sample, right? Primers are going to be added. So again, those are short DNA sequences that are specific and complementary to your target. So for us, that's SARS-CoV-2. And what's special here is the fluorescent probe that will release a light signal when more copies of your target sequence are made. And of course, we have the PCR components like nucleotides, polymerase enzyme, and these are going to come together to help build more molecules in the reaction. And then, of course, a buffer solution just to get everything in nice suspension and working well and happy.
Oh, this does remind me a lot of a PCR, traditional PCR reaction, but yes, I see those fluorescent probes. Definitely not present in a normal PCR condition. So that's how we know it's quantitative. So this is all happening inside of that thermal cycler machine that we talked about earlier. And the amplification actually takes quite a few cycles, and the target sequence copy number increases exponentially as the cycles progress. Remember, the unique part about qPCR, it's those fluorescent probes. So the more copies of the sequence in the sample, the more fluorescence will be detected. And this should all increase as the PCR assay proceeds through each cycle. So why is it called quantitative? It's because the fluorescence detection is happening at the molecular level. Every single molecule is detected and counted. So you can actually count up the number of fluorophores in your template at the end of the reaction and get a really accurate estimate, or I guess quantitation of your target.
Yeah, exactly. And so at the end of that reaction, after all the cycles are done, you're going to get a graph, and the graph is going to be a fluorescence detected. So remember, this comes from the amplified cDNA, which was converted from the RNA, which came from the virus that was in the sample collected from the patient. So let's look at the graph that's generated by the qPCR machine. Let's look at some sample data. So the x-axis on the bottom, that measures time, or PCR cycles, as the reaction progresses over time. The y-axis plots fluorescence detected at each cycle. So this is a measure of the abundance of your target sequence. So as you amplified, if your target sequence was there, if that SARS-CoV-2 sequence was detected, that fluorescence was going to shine, could be detected by the machine, and would be graphed as a point on the graph. So you'll also notice there's this little dotted line towards the bottom. That's a threshold of detection. And that's important to accurately distinguish any sort of background, what we call noise of the assay, from true signal. Because just having some reagents in the tube might cause a little bit of detection, but we don't want to make sure that that's very clear from a true signal. And that is why we have both positive and negative controls. These are important to include as references and to ensure that the assay was performed properly. So you have a known positive sample and a known negative sample that's included in your entire run. So for example, if in your samples there was no signal from the known positive control, well, then you know there must be a problem with the entire assay itself. If there was no signal just from your patient sample, but you got your signal from the positive control, then maybe you can go back and say there might have been something with the collection or the genomic extraction, the nucleic acid extraction, or maybe even the reverse transcription process from just that sample. You know that the qPCR itself was good. So true positive samples will always rise very far above that threshold of detection, and they'll follow that exponential curve, like Emily mentioned, reflecting that exponential amplification from that PCR process. And it shouldn't take very long to detect it, so fewer cycles in order to detect because it's very abundant. So the true negative sample, in contrast, may rise and fall as the PCR cycles, but really it should fall below or around the threshold of detection, because if it's truly negative, nothing should really be detected, so it should stay very low.
Okay, I'm really starting to see the importance of these positive and negative controls for clinical diagnostics, Christina. I thought they were important for the research lab. So remember, the key elements to this assay are the primers, and that's how we can detect the SARS-CoV-2 virus specifically and know that we're not detecting any other virus. And then we've got these positive and negative controls that are really going to help us distinguish and draw conclusions from the data that we're collecting. And something we didn't talk about was replicates. It's always good to run a sample more than once, and that's called technical replication. It's actually usually done 2 or 3 times to make sure that there were no technical errors in your hand pipetting, or perhaps a piece of dust contaminating your sample. We really want to make sure that, especially when it's diagnostics, that you have a really clear answer in your data.
Okay, so let's review and think of one scenario. We thought of one sample scenario here. So if Marcus ran 2 different samples, and here we're calling them sample 1 and sample 2, and generated the data shown in that graph here. Sample 1 looks positive because it's running right up there by the positive control. Sample 2 looks negative because it's running by the negative control. The data looks pretty clear. So let's think in a real world scenario, what could lead to these results? Well, I would think that sample 1 probably had viral RNA isolated at that first step. So from the swab, someone was infected with virus, the RNA was collected, the nucleic acid was isolated, the reverse transcription occurred, yielded a lot of cDNA, which was amplified and detected in the qPCR, right? But sample 2 might have had little to probably no viral RNA, so it wasn't detected in this assay. That's why it had really low results. Sorry, low signal.
I think I'm following your logic on that one, Christina, but I'm thinking of a different scenario that maybe could be happening as well. When I think about the viral replication cycle, I'm thinking maybe these 2 samples could be from the same person, perhaps at different times of the infection. We know now that when someone is exposed to the virus, there's an incubation period of about 5 days. That's 5 days where someone has the virus, it's replicating inside of them, but they don't have any outward symptoms. Then symptoms show up, which usually coincides with even more virus replication, and that's the infection period. Perhaps sample 2 in this example could have been collected on day 3 after exposure. And then sample 1 was collected on day 10, where more virus was in the person and could be detected by the test. What do you think, Christina?
Yeah, exactly. Timing for your test is key. And I kind of alluded to this at the beginning. If you have a known exposure, you'll want to make sure that you wait at least a few days, or at least take a few tests over multiple days to make sure you're truly negative. Because if you take a sample collection too early, it'll show negative, but the reality is that you have virus replicating inside and you need just more time for it to be detected. So if you would have taken a test later, it would have shown as positive.
Got it. So it's kind of hard to distinguish the difference between those 2. So it's basically taking, so viral load, Christina, tell me more about that.
Yeah, so, another idea that we had when we were thinking through this scenario is that, we just had the idea that those 2 samples could be from one person at different times in their infectious period, right? So what about if those 2 samples were from 2 totally different people and they were both infected? How could that happen? They could have a different viral load. So what does viral load mean? I'm gonna throw it back to you, Emily. Tell us what.
I hear you. So it's basically like the amount of virus, the individual molecules of the virus at exposure or inside of you. And someone can have a lot of virus replicating inside of them, but somebody might have less. And there's a range, and it's really variable inside of all of us as individuals.
Yeah, exactly. So as I learned about this test and as I really thought about the infectious period, about the virus and all these things, it really gets me thinking about negative tests and how they might not truly be negative. And it's not anything wrong with the test. It's just about when you collect the sample and drawing conclusions from that and what that means, right? So if you're positive, it's almost certainly positive. And we learned about how the test works, how the primers and the fluorescent probes and the qPCR, they have sequence specificity to that SARS-CoV-2 genome. So that molecular detection is going to be accurate. More likely than not, if you're positive, you're going to be actually positive. But if you're negative, well, you could still be infected, but the virus just wasn't detected. So again, it's that timing thing.
Got it. So the false negatives could happen if there isn't enough virus inside to be detected yet. All right, so maybe we can review for a minute. We learned that SARS-CoV-2 has an RNA genome, and that once it's isolated, that the viral RNA can be converted into cDNA, and then that can be amplified to be detected in the lab. And that gives us a quantitative readout of the virus that's present in the sample, and that data can be analyzed to give a final report on the status.
All right, so this is the exciting part for us. We get to open it up to your questions. So we did receive a few before we started, and we prepared a few slides. So we're gonna jump to those first. But any of you guys watching live, I know there are some because I see you, I see the numbers. If you have any comments or questions, don't be shy. Please put them in the chat and we're gonna get to them right after we do these slides.
Yeah, we'd love to hear from you. So we received several questions about why people experience different symptoms from the same disease. Well, let's think about it for a minute together. We know that our genetics, our genomes, they vary, right? We don't all have the exact same DNA. And we also know that we have, these unique differences in our bodies and immune response, and that could also be impacting it. We each also have different backgrounds and environments. We eat different things, and some of us might be battling other illnesses, or maybe we have, no access to healthcare or choose not to seek healthcare before symptoms progress. There's also lots of differences in the viral load at exposure, which we were discussing earlier. And there's also several symptoms that occur because our bodies are working so hard to fight the infection. You may have heard about the cytokine storm. It's sort of an inflammation that's associated with the natural immune system. So it's your body going into overdrive, accidentally overdoing it, trying to take care of this pathogen. So some of these symptoms can last a really long time. And as time goes on, we're learning more about this form of COVID, referred to as long COVID, and how to treat it.
So moving on to vaccines, of course, this is a huge topic right now as vaccine distribution is timely right now. We got a few questions, especially shout out to Julian from Maine. He asked about vaccines, and we're going to do a quick explainer here. We'll also highlight and encourage you to look at some really neat graphics by the New York Times written by science journalists, so you know that the information is accurate and trustworthy. And we have the link here and we can drop it afterwards as well. So let's go over the mRNA vaccines first. So how do these work? These vaccines deliver mRNA, or messenger RNA, of the SARS-CoV-2 spike protein into the human cells, into your cells. So this can directly enter the cell and use your body's cellular machinery in the cytoplasm to translate that message, that mRNA, and immediately make little spike proteins from this coronavirus. So your body is going to be making little spike proteins. That mRNA that was delivered in the vaccine, it gets degraded very quickly by the cell naturally. So the viral vector vaccines work similarly, but they need one extra step. So they contain DNA from the spike protein and an inactivated virus just to use as a vector to help deliver them into the DNA of your cells, into the nucleus of your cells, excuse me. So that DNA needs to enter the cell, and then it needs to get transported to the nucleus. So it won't affect your own DNA, but it needs the machinery that your human cells have in the nucleus in order to translate that DNA into RNA, transcribe the DNA to RNA, and then the RNA to translate it into a protein. So again, in this case, your body's gonna make little spike proteins, and the DNA and the viral vector used to deliver it from the vaccine will eventually get degraded by your cells. It takes a little bit longer, but it will get degraded by your cells. So in both cases, in the mRNA vaccines and these other viral vector vaccines, the newly made spike proteins will be recognized by your body as foreign. So your cells make these little spike proteins because of the virus, but your body's like, what are these? These aren't supposed to be here, right? So an immune response will be activated, and that is the start of how you make antibodies and how the vaccine works to help you build an immune response. So the next time, if your body encounters the SARS-CoV-2 coronavirus that has those spike proteins, it will have antibodies ready to fight it. Hopefully that doesn't happen and you don't get infected, but that's what these are designed for. So if you do have exposure, you will have the immune response ready and be able to fight.
Cool. Thanks for explaining that, Christina. This makes me think about that cytokine storm that I mentioned, and it's sort of an inappropriate immune response. Perhaps that's part of the goal of the vaccine as well, is to, mediate that for people. So when they do have a response, it's not, inappropriate. Awesome. Well, we've got one more question prepared here from Grace. Oh, I think we've got a couple, but Grace asked us about the pros and cons of each of the vaccines and whether there was anything special about mRNA vaccines in general. As Christina mentioned, the mRNA vaccines work a little more directly because the mRNA they deliver can immediately be used in the cytoplasm. It's fairly simple to design this type of vaccine in the lab, and the mRNA is quickly degraded. However, because the vaccine is RNA-based, the stability is an issue, as we've learned about today. Therefore, transporting and storing these at cold temperatures is a big cost, and it might not be convenient. While traditional viral vector vaccines are known to signal a stronger immune response, so far studies show the Pfizer and Moderna mRNA vaccines on the market are quite robust. So even though it's a new technology, they are yielding promising results 6 months out and maybe even more.
So it's also important to remember that all the vaccines prep your body with an immune response, right? So they give your body an upper hand at fighting the virus if and when it's encountered. You can still become infected, but you'll be able to fight it much faster and much stronger than without the vaccine. Also, depending on where the new variants rise, vaccines may be less helpful, kind of unclear, and more studies are needed. So the variants that you've probably heard a lot about, these are new mutations in the viral genome. These happen as the virus replicates and propagates in us. It's very natural for nucleic acids, especially RNA, like we learned how unstable it is, to get new mutations. So if these mutations happen to be in the spike protein, which is where the current vaccines are designed to target, then there's a chance that the vaccine will be less effective in that particular variant. If the mutations are somewhere else in the viral genome, then maybe the vaccines would not be as effective, or not be as affected by the mutation and be more effective. But you need a lot of studies in order to really understand how it's going to affect your body and the immune response. And studies are ongoing right now. So at least what we know currently, the vaccines are still very effective, and there should be no concern. It's better to get the vaccine, to have vaccination, and to have an immune response than to not, even though there are these variants out there.
Yeah, thanks for the explanation on that, Christina. I have been hearing a lot about the variants and thinking about it myself. And yeah, there's variation all the time, and only very specific variation is going to have an influence on this. Okay, so I think this is the final question that we have gotten in advance and prepared for you, and then we'll go to the live. So this one's about fighting COVID with drugs and therapeutics, and it's a great question. And so people want to know, how do you generally fight viruses with drugs? Again, we want to point you to that New York Times article. They have a really nice piece about different treatments that are available on the market today. So how do you choose which drug to use? Well, there's lots to consider. Think about how we learned about how the virus enters a host, replicates, and then transmits to the next person. Can any of those steps be targeted? Non-drug options like wearing a mask really help stop the transmission to the next person. And if you do have a drug, it needs to go through rigorous testing and approval to make sure it's safe and effective. Some drugs are easier to deliver than others, if it's a pill, for instance, versus an injection. And this can also affect who receives treatment. And you'll want to make sure that you're being, thoughtful and distributing this to everyone ethically. Other drugs in use now are antivirals, and these are used to block the virus itself from replicating inside of your body. Some new therapies include designing a modified receptor to block the virus from entering the cell. And there's a chance that giving an extra antibody can help your body fight after you've been exposed. All right, so thank you so much for your questions in advance. That was fun to be able to prepare some answers for you.
Yeah, that was awesome. Thanks for those that contributed questions first. I'm not seeing any questions pop up live now. I'll give you guys a few more minutes if you have anything that you want to say. I did post in the chat, if you're watching live, the link to this case study, plus some other genetics learning resources that we have. We have lessons, we have activities, we have presentations. So it's really a lot of content to fit all of your genetics learning needs.
And do not forget that this lesson that we gave today was just a short excerpt from that larger case study that we've developed. And if you're interested in learning a little bit more about the details of the COVID testing or those clinical lab careers that we had mentioned early on, make sure you check that out if you've got some extra time.
Yeah, and while we're here, while we have you, we'll share a few more things. So the Jackson Laboratory Genomic Education Division has a special STEM and undergraduate department, and that's what we are a part of. And so we focus on high school and undergraduate learning. And if you go to jax.org/STEM, I'll put that in the chat. You can check out all the different resources that we have for teachers and students at this level. And we're debuting today, actually, our virtual open house page. So this is a really cool new web experience for you to really dive into the lab without even leaving your home or your computer. So you can experience research talks, a lab tour, hear career chats, learn about what science is like after high school, all sorts of things that we have on a special site just for you.
Thanks for mentioning that, Christina. Yeah, there's a lot of great resources there. So we'll make sure to put that link in the chat so you can go check them out.
All right, well, if there are no other comments or questions, this was a lot of fun. Emily, I had a blast. It was really nice to go over this. And we really thank all of our viewers. You guys were great, at least just for joining us, even if you didn't have any comments. We appreciate your attention, and we hope you learned something.
Yeah, thank you so much, Christina. I had a blast chatting with you as well. And thanks everybody for joining us, and have a happy DNA Day.
Thanks everyone. Take care. Stay safe.